Later, they were called as nucleic acid by a scientist by name Altman. The DNA replication is called as semi-conservative DNA replication, as one of the parental strand is retained in the daughter cell. So what are the two very important types of nucleic acids, that is DNA and RNA, are the kinds of nucleic acid. Hello everyone, welcome back to yet another revision class of second PUC of chapter 6 called Molecular Basis of Inheritance. Moving on to the weightage of marks for this particular chapter. This chapter called Molecular Basis of Inheritance has a weightage of 10 marks. So it is a very big mark from examination point of view. So I'll be discussing here in this particular session revision class like what type of questions are going to be asked pertaining to this chapter called Molecular Basis of Inheritance. So the weightage of marks for this particular chapter is 10 marks, where you get the 5 marker questions will be of 2 questions. But again, as I keep telling you all, though it is a 5 marker question, but you will get maybe for 3 plus 2, that is 5, or 2 plus 2 plus 1 is equal to 5. So 5 marker questions, 2 questions you are going to get. But as I said, it might be 3 plus 2 or 2 plus 2 plus 1, or it might be total 5 marks question. So the weightage of marks for this particular chapter is how much? 10 marks. So let me move on to the chapter introduction. As you know, what you are going to study in this particular chapter, that is about the nucleic acids. As we all know that these are the organic macromolecules found in the nucleus of a cell, and they are acidic in nature, hence they are called as what? Nucleic acids. As you know, their DNA and RNA are called nucleic acids, which are found in the nucleus of a cell and show acidic properties, hence the name nucleic acids. The nucleic acids was discovered by Friedrich Mischer when he subjected the hospital bandage pus cells to certain digestive enzymes. One part of the cell remained undigested, and that undigested part he called them as nuclein, and that is how the word nuclein came into existence. So nucleus, nucleic acid. Then later they were called as nucleic acid by a scientist by name Altman. So nucleic acids were discovered by Friedrich Mischer, but the word nucleic acid was coined by Altman. So what are the two very important types of nucleic acids, that is DNA and RNA, are the kinds of nucleic acid. Deoxyribose nucleic acid and ribose nucleic acid are the two very important types of nucleic acids that are found in living organisms. As we know, the DNA is the genetic material, and deoxyribose nucleic acid, the three very important chemical composition of DNA is, it is made up of a pentose sugar called deoxyribose sugar, and the 4 nitrogenous bases, that is adenine, guanine, cytosine and thymine, and a phosphate group. So when it comes to RNA is also made up of 3 very important chemical composition, that is ribose sugar, then 4 nitrogenous bases called adenine, guanine, cytosine, and instead of thymine, it is uracil. So the thymine of DNA is replaced by uracil, and as of course it is having a phosphate group. Hence the name acid, nucleic acid. DNA and RNA are called nucleic acid. So you'll be studying the structure of the polynucleotide chain having a ribose and up the backbone and the base pairs, that is, as we know, according to the Ervin-Charles base pair rule, purines will never base pair with purines and pyrimidines with pyrimidines. So what are the 4 nitrogenous bases? A, G, C, T in case of DNA, and A, G, C and U in case of RNA. So A base pairs with double bond with T, and G or C base pairs with triple bond with C, or that is, or C base pairs with triple bond with G. Here A base pairs with double bond with T and C with triple bond, A base pairs with U. That is, in RNA, that is, the thymine is replaced by uracil. So it is U, and C with triple bond with G. So packaging of DNA helix, you'll be studying the search of genetic material in DNA, that is Griffith and Avery. As we know, Frederick Griffith was the one who conducted the transforming experiment in a bacterium called Streptococcus pneumoniae bacteria. So his experiment was based on Streptococcus pneumoniae bacteria. Then the properties of genetic material, that is DNA versus RNA, which is more stable. RNA, because DNA is, that is the thymine of DNA is more stable than the uracil of RNA. Hence, that itself will confirm that DNA is more stable. So, properties of genetic material you are going to study. Then the RNA wall, then the replication, DNA replication. As we know, the DNA replication is called as semi-conservative DNA replication, as one of the parental strand is retained in the daughter cell. So the machinery and the enzymes you'll be studying. Then about transcription unit. Anyhow, I'm going to discuss in the questions that what type of questions you are going to get pertaining to these concepts. Transcription unit and the gene, types of RNA, that is, we know there are types of RNA, tRNA, mRNA and rRNA, and the process of transcription where the genetic message from DNA is transferred to mRNA. So the genetic message from the transcription or the passing of the genetic information from DNA to RNA is called transcription, and thereon mRNA with the help of ribosomes makes a particular protein, and that is called as translation. Then genetic code. So what will be the important properties of genetic code? All that anyhow I'll be discussing in the questions. And why tRNA is called as the adapter molecule. Then translation, as I already said, from mRNA, the genetic message that is transferred from DNA to mRNA to a protein, that is called translation. So regulation of gene expression. Then the lac operant concept. So the regulation of gene explained with a lac operant, that is lactose, like switch on and switch off mechanism. Then human genome project, very important. And what are the salient features of human genome project? Then application and future challenges, that is excluding methodologies. And DNA fingerprinting, how important this technique is. For example, the applications of DNA fingerprinting, where you can identify the criminal who has committed the crime, may it be a murderer or a rapist, etc. And we also can solve the disputed parentage, like you can assign the parents to the lost children. And also when there are natural calamities like earthquake, for example, like a plane crash, fire. So in that point of time, it's very difficult to identify the bodies. So definitely this technology comes to the rescue. So all these are the important concepts what you are going to discuss in this particular chapter, which is very important from the board examination, that is from the examination point of view. Then what type of questions will be asked? As I said, though it is 5-marker questions, 2 questions are asked, but still, as I said, sometimes you may get the split questions, that is 2 plus 2 plus 1 or 3 plus 2. So let me discuss some of the questions that are pertaining to 1-marker question. What is transcription? The synthesis of RNA, that is mRNA, a template strand of DNA with the help of DNA-dependent RNA polymerase in the nucleus of a cell is called transcription. As I said, transcription is what? The genetic message that is transferred from DNA to mRNA to make a particular protein. So the synthesis of RNA, that is mRNA, a template strand of DNA with the help of DNA-dependent RNA polymerase in the nucleus of a cell is called transcription. Then moving on to the second question, as I said, why tRNA, transfer RNA? As I said, there are 3 types of RNA, that is tRNA, mRNA and rRNA. The very word itself will tell you tRNA stands for what? Transfer RNA. Why tRNA molecule is called as the adapter molecule? Specifically, why we use the word called adapter? Because it is an RNA molecule that transfer the amino acids to the site of protein synthesis into the ribosome during protein synthesis, hence the name adapter. So I repeat, it is an RNA molecule. tRNA is a type of RNA molecule that transfer amino acids to the site of protein synthesis. Which is the site of protein synthesis in a cell? It is ribosome. Into the ribosome during protein synthesis, hence the name, it is called adapter. Then why eukaryotic genes are called as split genes? As they have introns and exons, hence they are called as split genes. Next, 4th question, nucleotide ratio in RNA is highly variable. Give reason. It is a single stranded and there is no complementary base pairing, hence the nucleotide ratio in RNA is highly variable. Then 2-marker question, write the concept of central dogma. For example, if it is asked, the central dogma of protein synthesis is what? DNA to mRNA, mRNA to a protein. So the concept of central dogma proposed the unidirectional flow of information. So whatever protein has to be made, so that genetic message is passed on from the DNA, that is from the nucleus, the genetic message is picked up by mRNA. It comes out of the nucleus to the cytoplasm and helps in making a protein with the help of ribosomes. So that whole concept is called central dogma. So DNA to mRNA is called transcription, and from mRNA to protein it is called translation. Actually, this is called translation. Right. Now, what is the function of ligase? The small DNA fragments called Okazaki fragments synthesized on the lagging strand are joined together by the enzyme DNA ligase. So, what is the function of ligase? Ligase, as I said, the small DNA fragments called Okazaki fragments synthesized on the lagging strand are joined together by the enzyme DNA ligase. Then what is VNTR? The way expand, they might ask for expand. VNTR. What does VNTR stand for? V stands for variable, N stands for number, T stands for tandem, and R stands for repeat. So the non-coding DNA, which is also called as junk DNA, which does not code for any amino acid. So the non-coding DNA of an individual consists of repeated nucleotide sequences called VNTR. So what is VNTR or expand VNTR? Variable, number, tandem, repeat. So the non-coding DNA, that is junk DNA of an individual, consists of repeated nucleotide sequences called VNTR. Then what is, for 3 marker question, as I said, what is Griffith transforming principle? Explain. As I said, the transforming principle was discovered by Frederick Griffith in 1928. So what is this? Why it is called transforming principle, or what is transformation? Transformation is nothing but a kind of sexual reproduction in bacteria where genetic material of donor bacteria is transferred into the recipient bacteria without physical contact. I repeat, the transforming principle was discovered by whom? By Frederick Griffith in 1928. Transformation means what? It is a kind of sexual reproduction in bacteria where genetic material of donor bacteria transferred into recipient bacteria without physical contact. Next, why DNA replication is called semi-conservative? The very word itself will tell you. Semi means what? Half. Conserve means what? Preserve. So half of the parental strand is conserved or preserved in the daughter cell, hence it is called semi-conservative. So the parental DNA molecule produces 2 identical copies of daughter DNA molecules is called replication. What is DNA replication? The parental DNA molecule produces 2 identical copies of daughter DNA molecule is called replication. In both the daughter DNA molecules, one strand, that is one is the parental strand, which is also called as template strand, and the another one is what? Newly synthesized. So hence DNA replication is called semi-conservative method proposed by Michelson and Stahl. So one of the parental strand, half of the parental strand is conserved or preserved in the daughter cell, hence the name semi-conservative, which was put across by Michelson and Stahl. Then what is Chargraphs base equivalence rule? Explain. So Erwin Chargraph came out with a base equivalence rule, or Erwin Chargraph's rule, according to which adenine base pairs with thymine with double 2 hydrogen bonds and guanine will base pair with cytosine with 3 hydrogen bonds. So hence in a DNA molecule, the number of purines is equal to the number of pyrimidines. This is called Chargraph's base equivalence rule. So what is it? A, adenine base pairs with thymine with 2 hydrogen bonds, and G, guanine base pairs with cytosine with 3 triple hydrogen bonds. So hence in a DNA molecule, the number of purines is equal to the number of pyrimidines. This is called Chargraph's base rule. Now, enumerate the salient features of genetic code. So as we know, the genetic code what we are using right now is a triplet codon system. So what are the features of genetic code? Genetic code is universal. What do you mean by universal? The genetic code is common and found in all organisms. So genetic code is universal. Next, it is in triplet form, that is triplet codon. What is triplet codon? The genetic code is triplet code. For example, like AUG. So the using of 3 amino acids as a triplet code, which is coding for a particular amino acid like methionine. So it is usage of 3 nitrogenous bases coding for a particular amino acid is called triplet code. So the genetic code is triplet code, that is represented by 3 nucleotide sequences. The 3 nucleotide sequence present on mRNA strand that codes for a type of amino acids. Then initiator codon. The process of translation is initiated by the initiator codon AUG that codes for an amino acid called methionine. Then terminator codon. The process of translation is terminated or stopped by a terminator codon or nonsense codons which do not code for any amino acids. So, enumerate the salient features of genetic code. Genetic code is universal. It is triplet in nature, triplet codon, initiator codon and terminator codon. Then commaless. Triplet codon is commaless. What is it? The codons present on mRNA strand are translated or decoded continuously without stop unless it reaches terminator codon. So it is commaless. So the codons present on mRNA strand are translated or decoded continuously without stop unless it reaches the terminator codon. Next, non-overlapping. Each 3 successive nucleotides present on mRNA strand is a codon that codes for a specific amino acid. Hence, a latter or nucleotide of one of the codon never shares with another codon. Sensible. That is, a specific codon codes for a specific type of amino acid during protein synthesis. Degeneracy or redundancy. Few amino acids are coded by more than 2 codons. This phenomenon is called degeneracy or redundancy. All these are the features of a genetic code which is very important from examination point of view. Then, 5 marker question only. Explain DNA fingerprinting or DNA profiling. It is also called as DNA profiling and the applications of DNA fingerprinting. So, it is the technique to identify the similarities of the DNA fragments of 2 individuals, and it was developed by whom? Alec Jeffrey in 1985. So what is DNA fingerprinting? It is a technique to identify the similarities of the DNA fragments of the 2 individuals. For example, as I said, the wide application of DNA fingerprinting is to identify the culprits or the criminals who have done the crime at the site of crime. May it be a murder or it might be a rape, etc. So technique is very helpful to identify the criminals. Then DNA carries some non-coding sequences called repetitive sequences. That is what we call it as VNTR, that is variable number tandem repeats. Number of repeats is specific from person to person. So the size of the VNTR varies from 0.1 to 20 kb. Then repetitive DNA are separated from bulk genomic DNA as different peaks during gradient centrifugation. Then the bulk DNA forms a major peak and the other small peaks are called as what? Satellite DNA. There are 2 types of satellite DNA. Next, an inheritable mutation observed in a population at high frequency is called DNA polymorphism. What is polymorphism is what? An inheritable mutation observed in a population at high frequency is called DNA polymer, or it can also be called as what? Variation at genetic level. So polymorphism is higher in non-coding DNA sequence. Why? Because the mutations in these sequences may not have any immediate effect on individual's reproductive ability. That is the reason why polymorphism is higher in non-coding DNA sequence. And these mutations accumulate generations after generation and cause what? Polymorphism. So polymorphism plays a very important role in evolution and speciation. What is speciation? Formation of new species. Formation of new species. Formation of new species. So those are the type of questions which might appear in your board examination. So what will be the possible other type of questions which might be asked from the chapter called molecular basis of inheritance, like list the salient features of human genome project, or discuss lac operant model with the help of a diagram, briefly explain the process of translation during proteins. Either transcription or translation might be asked. Then for split question it might be asked, like why did Hershey and Chase use radioactive sulfur and radioactive phosphorus in their experiments? B, write the conclusion they arrived at and how they might ask, or enlist the salient features of the double helix structure of DNA which was proposed by Watson and Crick. So these are other possible questions which might be asked from the same chapter called molecular basis of inheritance. Moving on to the recap, or what are the learning outcomes of this particular chapter called molecular basis of inheritance. So at the end of this chapter, you'll be able to describe the structure of nucleosides and nucleotides, that is DNA and RNA. Then define transformation and describe Griff's experiment. Describe the experiment performed by Hershey and Chase to prove DNA and not protein is the genetic material, as they were the one who conducted experiment on Streptococcus pneumoniae. His experiments were based on Streptococcus pneumoniae. Explain mechanism of DNA replication, which is called semi-conservative DNA replication. Explain the process of transcription. Describe the salient features of genetic code. Explain the functions of mRNA, tRNA, and ribosomes in translation. Explain the regulation of gene expression and operand concept. Describe the Human Genome Project and the salient features of human genome. Explain DNA fingerprinting and its applications. So, experiment for genetic material. As you know, search for genetic material was first experiment done by Griffith material, that is taking Streptococcus pneumoniae, then Hershey-Chase experiment, then biochemical characterization of transforming principle. Then structure and composition of DNA, as we know, structure of a polynucleotide chain, DNA and RNA, that is a nitrogenous base, purines and pyrimidines. Pentose sugar will be ribose in case of RNA, ribose in case of RNA, and deoxyribose in case of DNA. A phosphate group, then nucleoside and a nucleotide. Nucleoside is a sugar plus 4 nitrogenous bases forms a nucleoside. Nucleotide, that is sugar plus 4 nitrogenous bases plus phosphate group, makes a nucleotide. Then structure of a polynucleotide chain, discoveries with respect to structure and composition of DNA. As you know, the double helical structure of DNA was proposed by James Watson and Crick, which is called as double helix model. And who was the scientist who discovered the nucleic acid? It was Frederick Mischer, who coined the term called nuclein. As I already said that when he subjected the hospital pus cells of hospital bandage with a certain digestive enzyme, one part of the cell remained undigested, and that undigested part he coined it as nuclein. Then Erwin Chargraphs rule, which says A plus T and G plus C is equal to when, where equal amount of purines base pair with equal amount of pyrimidines. Then enzymes involved in DNA replication, enzymes of DNA replication like DNA polymerase. Then we have helicase and DNA ligase. Then transcription unit, which consists of a promoter gene, a structural gene, and a terminator gene. Then types of RNA, as we know, we have mRNA, that is messenger RNA, rRNA, that is ribosomal RNA, and tRNA, that is transfer RNA, are the 3 types of RNAs. Then post-transcriptional, that is capping and tailing. Then proposal for genetic code was George Gamow, that is 3 nucleotides should make up the code. Then Hargobind Khurana, chemical method for synthesizing RNA with a base. Marshall Nirenberg was put across the cell-free system. Then Severo Okawa, enzymatic RNA synthesis. So proposal for all this was the very important concepts. Then structure of tRNA with an anticodon loop, like cloverleaf model of tRNA molecule, which has an anticodon loop, then amino acid acceptor, T loop, D loop, and variable loop. Then stages of protein synthesis, as we know, first step is initiation, then activation of amino acid, transfer of amino acid to tRNA, elongation of polypeptide, and termination of polypeptide. Then regulation of gene expression, that is at the transcription level, processing level, transportation level, and translational level. Then steps in DNA fingerprinting, as we know, DNA isolation, amplification, and digestion of DNA. Hope you have understood what type of questions might be asked from this particular chapter called Molecular Basis of Inheritance from examination point of view. Thank you.